Hi, I'm Heather James, conference producer at Technology Networks, and I'm here to introduce our next talk, Dissecting Developmental Origins of Infant Leukemia Using a 3D Model of Embryonic Blood Formation. Please join me in welcoming Dr. Denise Ragusa, postdoctoral research fellow at Brunel University London, where she also completed her PhD. Her current research focuses on experimental and computational approaches to generate clinically accurate models of hematological malignancies, aiming to uncover biological mechanisms and identify therapeutic targets. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. Without further ado, I will now hand over to Dr. Ragusa to begin her presentation.
Great, thank you everyone for joining and the opportunity to share some of our work on infant leukemia and a new model of embryonic blood development. So I'm based at Brunel University, and this is a collaborative project. So my talk will kind of encompass some of the latest developments in hematology and kind of embryonic blood development, imaging and characterization, and also some modeling strategies for both healthy and malignant hematopoiesis. So a great example of why spatial technologies and single-cell resolution is extremely important is embryonic development. So embryonic development is a very complex series of events that are required to go from a single cell to a complete organism. And you can imagine the single cell and spatial technologies are only now allowing us to understand all these stepwise processes that go from, just a few cells to complete organs and patterns that make up, human or mouse models. And just before, obviously, great efforts have been made to characterize these cellular populations and these processes using the available technologies. But it's only with a single cell resolution that we've been able to really characterize, what these cell populations are, how they develop and how they differentiate. And obviously, the advent of spatial technologies have further allowed us to really map how these transcriptional identities make up tissues and very complex patterns. And you can imagine if characterization of embryonic development is difficult, it's even more difficult to model these processes in vitro. And for this reason, technological advances are also needed to really put together multidimensional complex systems to truly mimic what is happening in vivo. So one example of one of such models is gastruloids, which are self-organizing 3D organoid models of gastrulation. So really early developmental processes that we can now mimic in, a 3D culture in a normal 96-well plate. So gastroloids are defined as self-organizing 3D aggregates. So they start from a normal 2D culture of embryonic stem cells. These could be mouse or human. And they will self-assemble in vitro. If you can see some of the original protocols, you plate these embryonic stem cells, and through several days of media supplementation, they will form these 3D kind of balls of cells that will exhibit specific patterns and developmental cues that mimics the developmental events that happen in the embryo. So they capture really early events, and more importantly, they are temporally and spatially accurate. So in fact, if you see some of the stainings of the original protocol by Van den Brink et al., you can see that these 3D aggregates are not uniform. They will have specific patterning of temporally and spatially accurate markers of different layers of development. And this will produce very beautiful 3D aggregates of cells, which again show specific axial patterning that will enable us to mimic all these developmental processes in vitro. And again, the setup of the cultures is a 96-well plate, so it is also very amenable to high throughput, downstream analysis. And ever since the advent of the first gastruloid protocol back in 2014, there have been many different adaptations of these protocols to try and mimic specific aspects of developmental processes. For example, there's been an advent of cardiac gastroids, gastroids to mimic somatogenesis, and different structures. And as we move on, these are getting more and more specific and adaptable to different parts of the developing embryo. So our lab is interested in a specific aspect of obviously the development, which is hematopoiesis, which is the production of blood. So we are obviously a little bit more accustomed to seeing hematopoiesis in this representation of a hierarchical representation of the adult hematopoietic system. So this is characterized by a hierarchy, as you see, with hematopoietic stem cells at the top that determine the differentiation into different lineages, myeloid, lymphoid, that then give rise to all different cell types that make up our blood and then eventually the immune system. And the hotspot site of hematopoiesis is obviously the bone marrow. And then we'll see these different differentiated cells. Some will reach obviously the bloodstream, and some of them might also be tissue resident. But that is really only applicable to the adult hematopoietic system. In fact, during embryonic development, this is completely different. So the way the adult hematopoietic system arises during development, it's more complicated than this. And I will come back to it later. So it has been challenging to understand adult hematopoiesis, which is obviously, as I showed you, a very complex set of different cell types. But more importantly, it is very dynamic, it's very heterogeneous. In fact, in the bone marrow, if we were to zoom in into the bone marrow context, we don't only see hematopoietic cells, but we also see a lot more different cell types that make up this microenvironment, including obviously the surrounding bone tissue, the marrow itself, which is made from a variety of different matrices and also non-hematopoietic cell types, such as endothelial cells, even some nervous system cells. So you can imagine how difficult it has been historically to understand, what different cell types have made and make up this complex system. And a lot of very advanced imaging systems through the years have really tried to map this complex set of different cells, which are not obviously static, but they change temporarily and obviously in response to specific stimuli. And obviously single cell and spatial technologies have really helped the hematopoietic community to truly map and characterize what a normal adult bone marrow would look like. And it is interesting, from single cell RNA sequencing, they've been able to reconstruct all these cell types that I was telling you about, and importantly, how well we now understand that the non-hematopoietic component of the bone marrow, it's really almost equally strong as the hematopoietic output. And more recently, actually one of the freshest papers that have come out, I'm now using also single cell RNA sequencing combined with spatial transcriptomics to map these cell types to where they reside within the tissue. And again, this is extremely complex from a technical point of view, because obviously the bone marrow system, it's spatially also complex to kind of create slides and really understand the spatial environment within the bone. And if it's been difficult to characterize and describe, this has also now resulted in a challenge to existing models of hematopoiesis. I showed you the classical, a little bit old-fashioned model of hematopoiesis, which is the hierarchical model, where again, hematopoietic stem cells sit at the top. And actually, through single-cell analysis of these populations and trajectories and cellular identities, we now understand that actually the hierarchical model doesn't really apply anymore, and the system is a lot more dynamic than we previously understood, and cell commitments, and in fact now the newest model, it's a commitment model, where different fates are decided in a system that is not hierarchical anymore, but it's more of a cascade of cellular identities and eventually what fate these cells will take. So if we go back to the developmental side of hematopoiesis, I told you how complex already the adult hematopoietic system is, but the developmental one has an additional layer of complexity because it is also a process that happens at multiple sites within the embryo. And interestingly, the very first waves of hematopoiesis are independent from HSCs, which again, in the classical model of adult hematopoiesis, sit at the very top and give rise to the different lineages after all. Whereas in developmental, and the first moment when blood arises in the embryo is actually independent from HSCs. And we characterize two different types of hematopoiesis. A primitive one, which is again the very first set of events that happens in the embryo, and then the definitive hematopoiesis, which is then what we will have for the rest of our lives as adults. And if you see, the emergence of HSCs only happens in the definitive part of the hematopoiesis. So actually, the whole of the primitive part is independent. And in the first wave, you will have, which happens in a separate location, which is the yolk sac. There is only an emergence of very primitive, kind of rudimental hematopoietic cell types, which are only needed for a very, let's say, early immune response. And after this, a second wave will arise, which is the precursor of our progenitors that then will make up the definitive hematopoiesis, which happens later on through the embryonic days and will migrate to the fetal liver. And this is the site when then most of the HSCs will arise and will then give rise to the complete hematopoietic system. And in particular, during the embryonic hematopoiesis, we have the involvement of a lot of non-hematopoietic tissues, especially in the very first primitive waves, which are necessary to then give rise to the very first hematopoietic-like cells. So you can imagine how precise this entire microenvironment, or actually microenvironments at different sites, needs to be for all these different cell types to arise at the right time in the right place. So the bone marrow already in adult hematopoiesis is complex. So the embryonic one has the added site of multiple sites. And again, not to go too much into the details of the cellular outputs of embryonic hematopoiesis, it's just important to, again, remember that the embryonic hematopoiesis begins in the yolk sac, then continues in the fetal liver, and eventually in the fetal liver, there will be an expansion of these hematopoietic sites, and that will give rise to then the bone marrow and the definitive hematopoietic system. So understanding all these different sites and the cells that make up all these systems has been very challenging. And without the resolution in sequencing and imaging techniques, reconstructing this process has been a true challenge. Now, as I mentioned, these methods are now allowing us to create an atlas of all these processes. And there has been a lot of effort from many different groups to truly map and create these atlases of embryonic hematopoiesis. And this has been particularly true at the fetal liver stage, because that's when you have the majority of cells expanding and are able to be characterized. Although this doesn't come without technical limitations. In fact, cell numbers are obviously really low, and the number of hematopoietic stem cells is extremely limited. So capturing that and really understanding the markers that can define different cell populations is still ongoing. But nonetheless, we are now able to truly understand what are the different cell types and their transcriptional identities, even at the spatial resolution, some of the fetal liver sections, which again, with advanced imaging, it really makes a completely different view that we had before of these processes. One of even more challenging than describing and capturing these challenges is if we want to have a model in vitro that captures all these different cell types and the spatial temporal precision of developmental hematopoiesis. And it has been difficult in the past to really establish faithful models because it is difficult to capture those multiple waves of the primitive and the definitive hematopoiesis. It is difficult to include all these different cell types and non-hematopoietic input, mainly the endothelial and vascular input, that needs to kind of cooperate and create these microenvironment of endothelial hematopoietic cells that then will give rise eventually to the hematopoietic system. And it's also been difficult to keep genetic manipulation to a minimum. There have been a lot of different models that have captured these different events through overexpression of transcription factors, for example, or supplementation with different signaling cues to give rise to these different populations. But it has been difficult to truly have a model that captures everything in the same place. So this is some of the work that our lab has been really interested in. How could we mimic all these processes in one place? So I'll come back to the gastrulloid system that I mentioned before. And we thought whether, the advantage of having these 3D assembling 3D structures could be modified to induce blood formation. So obviously the advantages would be that the cells will assemble themselves according to the natural, self-patterning events. There is a microenvironment that can capture those non-hematopoietic cell types. We can do this with minimal induction of different factors. And because it is in a 96-well plate, it's multiplexable and is trackable. So we used embryonic stem cells with a FLIC1 GFP marker, as you can see here in green, which is a marker of endothelium. So we can see the activation of this side of the gastrulloid. You can see that it becomes green around 96 hours in. So we modified the original protocol to extend to about 216 hours. And we added some specific cues to induce the formation of blood. So very early on, we induce an activin and chiron pulse that induce the BMP and Wnt to really start the promotion of the hemogenic endothelium, which comes slightly after with the addition of VGF and FGF2. And towards the late stage of the protocol, when we really want to boost the production of the late stage HSC's production, we add boosts of Sonic the Hedgehog, which will, again, promote the emergence of CD45 positive pre-HSCs. And then the very last 2 days, we add a cytokine cocktail of standard SCF, LIT3 ligand, and TPO, which are stimulating factors for hematopoietic cells. So with this setup, we were able to then induce cells from the embryonic stem cell stage into a self-assembling model of embryonic blood formation. So we characterized the model to be sure that it would capture developmental hematopoiesis from a cellular point of view by flow cytometry. We see that at 96 hours, we see the emergence of the FLIC1 GFP mark that marks endothelium. At 120 hours, we see the emergence of c-KIT positive cells, which is a marker for hemogenic endothelium. Later on, around 144 hours, we have early hemogenic progenitors characterized by CT41 expression. And we only see the hematopoietic marker CD45 which marks pre-HSCs or HSC progenitors at the very end of the protocol. So we also, with the use of single-cell RNA sequencing, we were able to cluster these regions, and the transcriptional identity of these markers does correspond to these populations that should be arising during the embryonic development. And interestingly, we also see non-hematopoietic cell types. As I said before, they are extremely important in the microenvironment structure of where these cells need to arise. And we see stromal and mesenchymal cell types and endothelial supporting types, which again reassures us that the hemogenic gastruloid protocol does capture that cellularity. And again, if we look up the markers again from single-cell RNA sequencing, we see that the endothelial markers are really highly expressed at early time points within the KIT positive population, which marks the hemogenic endothelium. And then at later stages, the CD45 population will exhibit all those erythroid and myeloid markers and progenitor identity, which again is further reassurance that the process is not only cellularly accurate, but also it follows a temporal sequence of events. And we also performed some immunofluorescence on sections of the gastruloids, and we can also see that they capture the topography of where these cells will be arising. And in fact, we see markers of KIT around these protrusions, mimicking the aortal endothelium covering. We see the 31 showing again the endothelial encasing, and CD45 cells, again the hematopoietic marker, stemming from these locations, which again are reminiscent of what we would be seeing in the region, in the embryo. So we also mapped the single-cell RNA sequencing from our gastruloids to established data sets from others, and we can see that our clusters do correspond to embryonic signatures in other data sets. And again, we see that some of our clusters have an expression of those early markers that mark hemogenic endothelium, and then late hematopoietic markers. So again, transcriptionally, also we see that the hemogenic gastruloid system does resemble the embryo. And then finally, we can also see that the output that we get from the gastruloids at endpoint, so at the very end, at the 216 hours, are able to form classical colonies. For those who are involved in hematology will recognize these at a frequency that is comparable to what hematopoietic progenitors would give rise to. And functionally, if we inject endpoint gastruloids into the adrenal gland of mice, we see an engraftment in both bone marrow and spleen. Okay, so just to summarize a very complex PCR, we have a tag in our gastruloids, which is a BFP fluorescent tag, and we are able to detect the marker of our gastruloids engrafting in bone marrow and spleen in these animals, which is absent from the controls, which were only injected with PBS. So this tells us that the cells that we are producing from this 3D model can also establish themselves into mouse tissues and maturate within these sites. So I'll give you a summary of this first part, which is the characterization of a new model of embryonic hematopoiesis. Again, I've shown you how complex the process is due to different waves happening in different sites. And really, the 3D reconstruction and single cell resolution is truly needed to understand how these processes work. And our model, the hemogenic gastruloids, is able to capture the cell populations, the topography, the temporal resolution, and even the functionality of the cells that go from the very early stages of embryonic hematopoiesis to the emergence of pre-HSCs. So this is a model that can be used for both normal and malignant hematopoiesis. And I'd like to give you some idea of how we can use this to model malignant hematopoiesis. Again, I'll go back to our hematopoietic hierarchy. So hematological malignancies are obviously cancers of the hematopoietic system. We normally call them leukemias, and they can really affect any cell type within this hierarchy. And different lineages will give rise to myeloid or lymphoid leukemias, depending on which lineage is affected. And the cell of origin really refers to what cell is hit by these malignant events that will give rise to these leukemias. And the leukemias can be both defects in proliferation or differentiation of these cells. Leukemias are generally a disease of the elderly, but they can also affect children. And you can see, if you look at some epidemiology in the UK, you see that some leukemias peak at different age groups. So myeloid leukemias, again, tend to peak at later in life. However, there is one peak in the infant group of 0 to 4 years old. Whereas acute lymphoblastic leukemia, so from the lymphoid lineage, seems to be prevalently in the very early childhood, and then doesn't really come back in later life. What is interesting about leukemias, it's that there are a lot of different genetic and molecular features, and there is a lot of difference between subtypes affecting different ages. So again, our lab is interested in pediatric acute myeloid leukemia. So I'll give you a very interesting graph to show how different the proportion of different molecular subtypes is between infants, which are defined as under 2 years of age, and older children, 2 years onwards. So you see that some of them might be in common, but some subtypes are unique to this age group, which means that there is something about infant presentations of this disease that is very different. So presumably the biology is also different. And we're particularly interested in this yellow size of this pie chart, which is the translocation T712, which again is absent from any other older children and older adults. So there is something really unique about this subtype. So this translocation is very rare, again, affects infants between the age of 0 and 2. There is a fusion transcript, but this can only be seen in half of the patients, so it doesn't seem to be necessary for the leukemia itself. What seems to be in common with all the patients is the overexpression of the MNX1 gene. So MNX1 is one of the genes involved in this genetic exchange. And what is even more interesting is if you overexpress MNX1 in adult cells, it won't be able to produce any leukemic transformation. So it seems to be only affecting a very specific time window, a developmental time window. And very recently, it has been proven that this leukemia originates in utero. So already during embryonic development, there are events that would then lead to the full leukemic event taking over in the newborn. So our lab has really looked at what makes T712 different from other leukemias. We looked at a lot of bulk RNA sequencing. Again, this is what is available so far to these leukemias. And you can see, if you look at whole transcriptomics, that 712 patients have a very distinct transcriptional profile compared to the rest of childhood leukemias. So there is something unique about it. We also spent some time looking at different signatures that could define T712 compared to AML. And we derived these signatures, and they have a lot of enrichment of adhesion, kind of biological processes, and morphogenesis genes. And if you look at the cell type enrichments, they are very early embryonic cell types. And again, which could explain the in utero origin. So far, there have been a number of models that have been created to try and mimic this T712 AML in vitro. We've had MNX1 overexpression approaches both in mouse and human setting, and we also have an in vivo model recently by transplantation of fetal liver cells into mice, and also some CRISPR attempts from us and others that have been able to really engineer the translocation into the cell using CRISPR-Cas9. All these models have really elevated our understanding of T712, although they are all in different type of settings, and we haven't found yet one model that is able to fully recapitulate what is going on with this leukemia. And one of the reasons might be, and if we really go back and reflect on the clinical manifestations of this leukemia, is that, again, it only hits really young children. It originates during pregnancy. The transcriptional features that we see hint that the cell of origin might be a very early developmental cell type. And again, we know that MNX1 cannot transform adult cells. So we started thinking maybe our hemogenic gastroloid model might be providing that context, the embryonic context in which MNX1 works within these young children. So we decided to, again, make use of the gastroloid model by lentivirally engineering an MNX1 overexpressing mouse embryonic cell line, and we assembled gastroloids. And we compared to a normal empty vector control. So you can see that MNX1 overexpressing gastroloids assemble properly. You can see that the green FLIC1 GFP marker is expressed, so there are no major defects, although you can see that the MNX1 overexpressing one are a lot larger, both in size and in cell numbers. So we already see that MNX1 might be doing something in this system. So the advantage here is obviously that we can really reconstruct the events of MNX1, not only at the cellular level, but also in, within the context of the microenvironment that they would be acting in vivo. So not to go too much into the details of the features, we see that the effects of MNX1 seem to, on the hemogenic development, seem to hit at 144 hours. So this is the time when from the yolk sac, we move into the hemogenic endothelium and we start seeing the first hematopoietic cell types arising. So it's a very early progenitor. And we see an expansion of c-KIT positive cells, again, a marker of hemogenic endothelium. And if we look at RNA sequencing between the 144-hour and 216 hours of the MNX1 overexpressing cells, we see a lot more early hematopoietic regulators being upregulated at 144 hours rather than later on. And when we kind of deconvolute the RNA sequencing using our single cell data from the gastroloids, we see that MNX1, so T712 patient transcriptomics map within the same kind of transitional identity than gastroloids with MNX1 overexpression at 144 hours more so than the latest stage at 216. And just to put this into context, we also took some transcriptional profile from another leukemia within the same age group of the KMT2A group, which gives rise to a more mature leukemia subtype. And in fact, this one maps at clusters which are more differentiated and towards the very end of the time point at 216 hours, which again allows us to really dissect the cellular population at the time point at which MNX1, also in comparison to patient transcriptional programs, are more likely to be leukemogenic. So with all these analyses taken together, it is most likely that MNX1 might be hitting a very early developmental progenitor, which is only present within this, this temporal window. So when we take these gastroloids, disassemble them, and put them into methylcellulose assay of leukemic transformation, we see that they both transform, both at 144 and at 216. However, when we look at the transcriptional features, only the colonies from the 144 hours truly capture the cellularity that we see in the patients, and also the morphology of really early undifferentiated cell types that we would be seeing in the clinic. Again, fully understanding, how MNX1 might be acting in different contexts. So again, the capacity to transform is still maintained, but the resemblance to patient's features is lost. So, and again, this really proves that MNX1 might be having different effects in different cell types, and having the right embryonic context can really give us a platform to understand how this translocation gives rise to this leukemia. So just to sum up both sides of the talk, both of the hemogenic gastroloid system, again, we already saw that having a 3D spatially resolved and temporally resolved system can really help us understand all those processes of developmental hematopoiesis, but also we can use it to understand when these processes go wrong. So in the case of infant AML, this can really help us dissect the origins of these malignancies, which trace back to the in utero life rather than newborn life. And we have seen some of the effects of MNX1 targeting a really early progenitor, and that we can model what we see in the patients within an in vitro system. And with this, I would like to thank everyone who participated in this project. This is a truly collaborative project across different institutions in Europe. And this is the bioRxiv preprint associated with this work. And with this, I'd like to thank you for your attention, and I'm happy to take questions.
Thank you so much, Dr. Ragusa, for your fascinating presentation. We have already had some excellent questions come in for you, so I'm going to start with the first one right away. And that is, how can spatial transcriptomics be applied to this work?
Yeah, so we are currently characterizing the hemogenic gastroloid model first to understand. So we've done some work with immunofluorescence, but now the spatial transcriptomics with sequencing will really allow us to apply the single cell data that we have within the 3D structure of the model. And in terms of the leukemia, this will also be very interesting because we can see how leukemias can change perhaps the spatial interplay of the cellular populations that are affected.
Excellent, thank you so much. Another question that's come in for you, what is the biggest challenge of single-cell omics for leukemia research?
So I think the biggest challenge, I would say definitely cell numbers. It is difficult to sequence enough numbers to, at least at this moment, to really have a population understanding of the leukemic populations compared to the healthy ones. And I think, again, at the moment, we are unable to do true spatial transcriptomics at the single cell level for leukemias, because compared to solid tumors, leukemias are liquid, obviously. And while in the bone marrow, we can get an understanding of spatial resolution, in the bloodstream, this gets more complicated. So we don't really know yet how to solve that problem.
A great answer. Thank you so much. We've got time for a couple more here, so I'm going to hop into the next one. Are next generation sequencing technologies already in use in clinical practice in hematology?
Yes, so single cell, not quite, but we are getting more and more, obviously, with the cost of single cell technologies, getting lower and lower, and clinical validation, it's truly bringing them to the clinic. In terms of next generation sequencing in general, many patients have now NGS assays routinely, especially more challenging cases, to really understand what the molecular subtype is. So short answer, yes, but at the single cell resolution and spatial, there might be still work to do.
Lovely. Thank you so much. And I think we've got time for one final question here, and that is, what is the advantage of drug testing in a 3D model?
Yeah, that's a very good question. I think a lot of these 3D organoid models are heading towards a drug testing side of things. Obviously, for us, at least in our model, we have a 96-well plate setup, which is already how most drug testing work in vitro. The additional advantage of it's obviously that you're seeing the effects of the drug within a pool of cells rather than just a single 2D culture. So that way, we can also understand potentially the effects of the drug on non-malignant cells, and overall, if there might be some effects that we didn't really foresee in the 2D cultures.
Lovely. Thank you so much, Dr. Ragusa, for your insightful answers. Unfortunately, that's all that we have time for today, but we appreciate you taking the time to share your expertise with us. Any
Thank you.
questions that we didn't have time to get to will be sent to the speakers after the event. As with all other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks once more, Dr. Ragusa, and we'll be back soon with the next talk.